Hello, good whatever time it is, wherever you are. My name is Juliana DiGiacomo. I work with Rutgers University, and I'm going to be discussing the environment as a driving force in the coevolution of Ferredoxin termini. But first, we are going to take a step backwards to really contextualize the significance of this claim. Biological redox reactions are catalyzed in nature by a set of enzymes called oxidoreductases. This grouping can be separated into 6 major subsets, which I have included here, each with really diverse redox functionalities. The origin of oxidoreductases likely parallels with the origins of life itself. However, the actual path of this evolution is largely unknown because of the immense structural and functional diversity that I've outlined here. Herald 2014 suggests 10 distinct origins for oxidoreductases where all iron-containing oxidoreductases share a single origin. This is especially significant as this group constitutes nearly 50% of all metal-containing oxidoreductases. So now we are just focusing on understanding the origins of these iron-containing proteins. There are numerous protein folds which contain these iron-hosting domains, and the most ancient of which likely looked like extant ferredoxin. Ferredoxins are a subset of electron transfer proteins which contain iron-sulfur clusters stabilized by assisting binding motif. Therefore, the study of the origin of ferredoxin serves as a proxy for the origin of oxidoreductases in general. Ferredoxins function using a critical iron-sulfur cluster. Life originated on Earth when the levels of oxygen was limited to completely absent, which permitted metals, including iron, to exist as soluble compounds in the ocean, such as these iron-sulfur clusters, which really could have formed spontaneously. Also supporting the antiquity of ferredoxin is that the ferredoxin binding fold is highly evolvable, derived from the low hydrogen bond energy per residue. This, along with the fact that ferredoxins are also ubiquitous to all life, supports that ferredoxin is also very ancient. In terms of how this ferredoxin evolved, it is accepted that at one point, the gene of some ancient iron-sulfur redox protein was duplicated, leading to a symmetric protoferredoxin, which I've outlined at the bottom of this slide. However, extant ferredoxins are asymmetric iron-sulfur proteins. At this point, each half could have acquired random mutations, which led to modern ferredoxin, or the evolution of each half could have been driven by some external force. Previous phylogenetic analysis with my lab of the amino acid sequence of each independent terminus of ferredoxin displays significant sequence conservation unique to each half. This suggests that the two halves of ferredoxin evolved independently under the influence of some external factors that specifically drove certain sequence changes to be conserved in each half. Thus, the objectives of the study is to directly to understand the external forces which drove independent coevolution of ferredoxin and therefore oxidoreductases. But more broadly, we hope to identify the basic factors of electron transfer and contribute to establishing the possible conditions for the origins of life. Of course, central to the study is the hypothesis. In this study, we suppose that following a gene duplication event, environmental factors drove the independent coevolution of each terminus of ancient ferredoxin. We specifically chose to start by looking at the impacts of temperature and pH, because not only are these some of the most simple factors of environment, but consistent with the hypothesis that life originated at a deep sea hydrothermal vent, temperature and pH are two characteristics of the environment of early life, which would have drastically changed as life diversified from that point. And moving forward, for the study, it was most advantageous for us to focus on the most ancient group of ferredoxins. And we find this in 4-iron 4-sulfur cluster ferredoxin specifically in methanogenic archaeal host organisms. So moving on to methods and results. In order to analyze independent coevolution, we had to split each ferredoxin into its respective termini. We did this with a novel methodology to my group by locating the halfway point in protein 3 structures and comparing this to an alignment with all relevant ferredoxin sequences, which were analyzed in this study. This created an N-terminus and C-terminus for every protein. Finally, we arranged these termini into maximum likelihood phylogenetic trees, which allowed us to analyze the evolutionary relationships between these proteins. Here you can see the initial phylogenetic tree, which was generated from the aligned Ferredoxin termini. The same termini are generally clustered together, indicating that they are similar across different proteins. As you can see in the bottom, highlighted clade. However, above this, you can see that there are several clades that are mixed, showing that the termini still display a level of sequence similarity within the same proteins. When we consider host optimal growth temperature data, it appears that in the instances where thermophile and hypothermophile sequences appear to be deeply branching in the clade, which means they are closer to the origin, the sequence is typically from the C terminus of Ferredoxin, which I've outlined here. You can also see that the thermophile and hypothermophile hosts are generally clustered together, whether they are at the closer to the origin or elsewhere in the tree. Unfortunately, no such correlations existed when pH data was overlaid with termini data. Further investigation was focused on a large clade that was almost entirely composed of Ferredoxin N termini. This is highlighted in blue on the slide. The sister C termini of the blue clade formed 4 distinct clusters, the sister alpha, beta, gamma, and delta. While there are no differential distribution of pH among these sister clades, we do see a pretty clear-cut distribution with temperature, where especially sister clade gamma is really dominated by hypothermophiles and thermophiles. To investigate further, multiple sequence alignments were compared between the termini of each sister clade. All alignments were visualized as sequence logos, visualized on the slide here. Each sister clade contained residues which were differentially present, but the most notable was in sister clade gamma, which is dominated by thermophiles and hypothermophiles that I mentioned before, which contains a unique and really heavily conserved valine at position 5 within this termini, and a tripeptide segment. So now, what does it all mean? The real takeaway from the study is that these findings suggest that the Ferredoxin termini did likely co-evolve independently, where temperature was one of the driving factors for residue change in each terminus, and pH, as far as we know, does not appear to be in such a direct way. The C termini sequence also appears to be more ancient than the N termini, and perhaps reflects the proto-Ferredoxin, which underwent a gene duplication originally. Within the C termini, a valine near the beginning of the sequence appears to be critical to thermophile and hypothermophile Ferredoxin. We also know that an ancient Ferredoxin likely evolved near the origins of life and has diversified into all iron-containing proteins, including 50% of all metal-containing oxidoreductases. Therefore, this investigation also suggests that environmental temperature was a driving factor in the ancient evolution of life's really earliest oxidoreductases and proteins that were capable of electron transfer. As thermophile and hypothermophile sequences appear to be deeply branching, the study also supports that life's earliest oxidoreductases developed in host organisms with extremely hot environments. These observations aligned with previous studies on small subunit rRNA and, of course, the hypothesized origin of life at superheated hydrothermal vents. And that is all I have for you today. I would like to acknowledge my head of my lab, Dr. Vikas Nanda, and everyone else in the Nanda lab, the Nanda Dream Team, and everyone else who's helped with collecting source materials and getting this project done. So thank you.